Saliva is largely produced by three major pairs of salivary glands that lie outside the oral cavity and discharge saliva through short ducts into the mouth. If we zoom in on the parotid gland, we can see that it resembles a cluster of grapes. It's composed of hundreds of tiny lobes called acinus that produce saliva, which are connected by a duct that collects the saliva produced. Acini are composed of two different kinds of cells, serous acinar cells, which secrete proteins in a solution with a high concentration of ions known as isotonic water, and mucous acinar cells, which secrete mucin proteins, giving saliva its viscous and sticky characteristics. The acini can be composed of serous cells, mucous cells, or a mixture of both. The secretory acinus lobes merge into intercalated ducts, which are lined by a simple low cuboidal epithelium and are surrounded by myoepithelial cells. These ducts continue as striated ducts and have a folded basal membrane to enable active transport and enhance the absorption of ions. The striated ducts lead into interlobular excretory ducts, which are lined with a tall columnar epithelium. To produce saliva, cells utilize a specific mechanism to transport water from the capillaries to the core of the acini. However, controlling the movement of water is not a simple process as there is no protein capable of actively pumping water across the cell membrane. Water enters and leaves the cell through specialized proteins called aquaporins, which act as channels facilitating the free passage of water. In order to control the movement of water, cells adjust their water potential by altering the ion concentration both inside and outside the cell. To extract water from the capillaries, acinar cells uphold a higher intracellular concentration of chloride ions in comparison to its basolateral side, elevating the water potential within the cell, and when the acinar cells open chloride channels on their apical side, chloride ions are discharged into the salivary ducts, drawing water into the salivary ducts. To build up a high concentration of chloride ions, the acinar cells possess chloride cotransporters that utilizes the electrochemical potential energy of sodium and potassium ions to transport chloride ions against their electrochemical gradient. This cotransporter introduces 2 chloride ions for every sodium and potassium ion. The energy required for this process is derived from the electrochemical potential provided by the sodium-potassium pump, which utilizes the chemical energy of ATP to sustain it. Another kind of chloride cotransporter utilizes the electrochemical potential of ionized bicarbonate to transport chloride ions into the cell. The bicarbonate concentration is maintained by the enzyme carbonic anhydrase, which facilitates the rapid interconversion of carbon dioxide and water into bicarbonate ions, which serves as an electrochemical force that aids in the transport of chloride ions. To release chloride ions through the apical side of the cell is a slightly more complex process, as it is regulated by parasympathetic neurons that establish synapses with acinar cells. These neurons release acetylcholine, which acts as a ligand on muscarinic receptors, activating G proteins. The activation of the inositol 3-phosphate signaling pathway is subsequently triggered, leading to the release of calcium ions. The calcium ions, in turn, open chloride channels on the apical membrane of the cell, thereby regulating saliva production. The production of salivary proteins is also regulated by the nervous system. Acinar cells establish synapses with sympathetic neurons that release adrenaline. Adrenaline acts as a ligand on adrenergic receptors, which in turn activate a specific type of G protein. This activated G protein initiates the cyclic AMP signaling pathway, leading to the activation of pKa, a kinase responsible for activating motor proteins involved in vesicle transport. When the vesicles reach the cell membrane, they undergo fusion, releasing the salivary proteins into the salivary ducts, such as mucins, amylases, defensins, cystatins, histidins, lactoperoxidases, lysozymes, lactoferrins, and immunoglobulins. Some of them begin digestion by breaking down monomers of polysaccharides, others function as biological weapons against bacteria, viruses, and fungi, and others serve to lubricate food. Newly produced saliva contains a high concentration of ions. To prevent ion wastage, cells in the intercalated ducts passively absorb these ions by utilizing sodium and chloride channels located in their apical and basal membranes. The ions move from areas of high concentration to low concentration naturally. However, to ensure the collection of all the remaining ions present in isotonic saliva, striated duct cells employ chloride bicarbonate cotransporters. These transporters facilitate the movement of ions from areas of lower concentration to higher concentration, resulting in saliva becoming a hypotonic solution. When you indulge in something delicious like tacos, it is common for overstimulation to occur. In such situations, the parasympathetic neurons are activated and stimulate the myoepithelial cells surrounding the acini and striated ducts. These cells contract, causing a rapid expulsion of saliva from the salivary ducts. The saliva is released so quickly that there is insufficient time for the absorption of chlorine ions, resulting in the saliva being expelled in its isotonic form. However, these chlorine ions are not wasted since they eventually reach the stomach where they play a role in the production of hydrochloric acid.